Hey everyone, my name is Tiffany and I will be doing a presentation today on what I do. So this is a pre-recorded version, just because we never know with load shedding what's going to happen. And so if there are some glitches, I've never recorded on Zoom before, so if there are some glitches, I do apologize. But hopefully this is a seamless experience and I hope my screen is sharing with everybody. So I am just going to start off by talking a little bit about myself and then what I do, and yeah, that's pretty much what this presentation is about. So last time I did this talk was at UCT, and they all found it really interesting because, you know, archaeologists don't really delve into paleontology all that much when we're busy doing our studies. We do go into it with our human evolution, but not in great detail. And then the time before that I did this presentation was to my nephew's grade 4 class, and they're really easy to impress because I just show them a fossil and everything's a dinosaur. So that was a really, really easy presentation to do for them, and they loved it. So I thought that was quite cool. So I'm going to start off by just talking a little bit about myself and how I got into this field. And yeah, so I graduated from UCT in 2013. I did my honours in archaeology there. And then I went on to Stellenbosch University, where I did my NRF internship. And there, that was kind of cool because we got to learn a lot about, well, I got to learn a lot about CT scanning. So basically what I was involved with was a project on the CT scanning of mummified animals in our South African collection, which is primarily sitting at Iziko. We've got a massive collection of mummies, well, animal mummies, and also just Egyptian stuff in general. So that was really, really cool. It was really interesting. The whole idea is non-invasive research. So here is just an example of the kind of things that we did. This was all done at Stellenbosch University's CT scanner. So bottom left, you can see there's a picture of a falcon and the CT scans and the different levels of what you can see inside, which was really nice. It meant we didn't have to touch the specimens, we didn't have to unwrap them or anything like that. And another thing I was involved with there was this picture that's currently on the screen is actually a CT scan of what was inside one of the mummies, which was very cool. And so you can actually see, let me just get my laser pointer, there we go. So you can actually see that there's a jaw there with some teeth, and involved with was trying to identify what this was that was inside. So, yeah, that was really, really cool. And then, how do I get my pointer to turn off? Well, anyway, so that was one of the jobs that I did, and I did that for a year until 2015, and that was really fun. That was so super interesting. I got to learn a lot about genetics as well, because I was based in the genetics department, and that's also super cool. So yeah, then I started working at Iziko at the end of 2015, and I went straight into the paleontology department. But I was involved in a lot of archaeological things while I was there. I organized and hosted a human evolution workshop for teachers, and I also did a human evolution workshop for grade 12s and sometimes grade 11s, but they mostly learn about in grade 12. So I was still involved in archaeological things and human evolution, which I also find super fascinating. So yeah, then I was also involved in a cleanup at the fossil park in Langebaanweg, yeah, that sort of area, Langebaan area. And some of you might find this, or might see this is quite familiar because the field school, the third year field school used to be hosted at the fossil park as well. And so I remember spending 3 weeks of my life in third year at this place. But the idea behind this is that this dig site is outside, it gets covered in a lot of dust. So what I was involved with there was just cleaning up the specimens. So here you can see on the left, again, is a fossil that is covered in dust, and we were cleaning it up. So that's what it ended up looking like on the right. And this is done on an annual basis, and I think even every 6 months now. So yeah, then at the end of 2019, I left the museum and started my own little thing. So when I started at the museum, I was still fully, you know, archaeology, and within my first week there, I went straight into a field trip. So that was a very, very interesting learning curve because I didn't know anything about paleontology, or at least Karoo paleontology at that point. And the way that they work in the field is very different to how archaeologists work. But don't worry, paleontologists are just as weird as us wonderful archaeologists, and they love their whiskey just as much. So here is some photos, just I think it was the last night there or something, we had a little party and it was really fun. So yeah, just to demonstrate how archaeologists and paleontologists aren't that different after all. So just a quick intro into paleontology and where the Karoo fossils come from. I know Claire Browning did a presentation not so long ago, and she probably went into a lot of detail about this stuff. So I'm just going to quickly breeze over this because I actually do work for her. So yeah, I'm sure she explained this beautifully. So hopefully I do it justice as well. So this is the history part of my presentation. So we learn about deep time in archaeology, but we can only really kind of imagine life from the last couple of thousand years. Maybe we can comprehend a couple of 10,000 years, but the stuff that we work on is not only millions, but it's hundreds of millions of years old. So our focus is between the Permian and the Triassic. There's an extinction event that happens there, and you can see I put nice bright yellow arrows so that you can see exactly where we're talking about. So in other words, a really long time ago. And obviously the world looked very different. We're looking at 250 million years ago, so something like down here at the bottom. And you can see Africa was joined against Brazil, and yeah, everything looked really different. So there are massive fossil deposits in South Africa. There are also huge fossil deposits in Russia, China, Brazil, and Zimbabwe that all have very similar species. So that really proves how the land has moved around. And yeah, so the Karoo Basin is one of the best places on Earth to study the effects of extinction, and on the biodiversity of land-living vertebrates. So 96% of all species died during that extinction event, and the Karoo was a very different place back then. It was lush, swampy, green. There's a massive diversity of animals there, so yeah, so it looked very different. So what most people think of, so that's the background, sorry, of Karoo paleontology, and specifically we're working on Karoo paleontology. We do have other deposits in the Eastern Cape. I'm a little bit involved in that, but I'll get into that a little bit later. But for the most part, just the Karoo stuff. So when we say fossils, most people think of dinosaurs. So like these beautiful creatures. So Triceratops, and Brontosaurus, Brachiosaurus, and obviously T-Rex is also a big one that everybody thinks of. So these are all the kinds of animals that everybody thinks about when you say, oh, paleontology or fossils. They immediately think of dinosaurs. However, I think the stuff from the Karoo is way cooler because it's, and then obviously this is a display of Iziko, of the two dinosaurs from Africa as well. But in Karoo paleontology, we have some, and very famously Archaeotrics, which a lot of us are familiar with as well. So therapsids are the things that I primarily work on, and the species is a mixture of mammal and reptile, so we call them mammal-like reptiles. They've got all sorts of different traits. So this is what one of them looks like, a really large specimen that is on display at the museum at the moment. And then you also get Lystrosaurus, which is a little bit smaller. You also do get large ones as well, but yeah. Then this is Diectodon, which is quite a common, it's kind of like the size of about a Dussie sort of size. And yeah, very cool species as well. They used to do really cool burrows, which is I can't remember the name of it right now. So they used to do these really cool curled burrows. And then, yeah, we also get our, not Youngina. Well, anyway, so the reason why these are called mammal-like reptiles is because they had a pineal foramen on the top of their head, which indicates that they were able to regulate their temperatures. So that means they could have been cold-blooded. They also had occipital condyles, which allows for the articulation, so they could move their heads this way and up and down as well. And they also had a secondary palate, which meant that they could eat and breathe at the same time, which is a mammalian trait as well. It also could indicate that they were suckling. So yeah, then this is one of my favorite specimens. It's a gorgon. These species, yeah, they have the most amazing teeth. Like, I just think they're so stunning. Alrighty, then that is just a background on the types of fossils that we get in the Karoo. I just want to talk a little bit how in the field it's kind of different to what we do in archaeology and just how it differs, and then I'll get into what I actually do. So, field work to archaeology, I was thrown in the deep end because first week I went into the field. Instead of setting up beautiful squares, when you go for paleontological field work, you go out into the middle of nowhere, as this picture demonstrates, and you walk and you look, and it is so different. So what we look for when we're out there is we look for these really steep slopes that have quite high erosion rates as well, so they can expose the fossils, find fossils. And basically what you're doing is you're looking for things that look a little bit different. So here's another picture demonstrating that you're in the middle of nowhere, but the shades, well, yeah. So here is a demonstration. So this is part of the mystery of my presentation. So in this picture, we have a fossil. Now you end up, well, just to give you an idea, you end up walking between 5 and 8 kilometers or so every day. You're walking, you've got all your tools in your bag with you, you've got a geological pick, you've got paraloid, which is a way to coat the fossils that will keep them protected when you find one. So you walk and you look for things that look different. So like I said, in this picture, we have a fossil. It's quite difficult to see where that fossil is, of course. So this is a slightly zoomed in picture of the previous one. And again, there's a fossil somewhere in this picture. So I will leave this here for a couple of seconds, see if maybe you could spot it. But it is quite tricky because it all blends in pretty well. So in case you haven't spotted it yet, here's an even further zoomed in picture. And again, I promise there is a fossil in this picture. Okay, let's see. So then, let's leave the pointer there. All right, then an even more zoomed in picture. And here you can see this rock here is very slightly different to everything else that is around it. So a little surface find of a skull, and when you turn it over, oh no, this wasn't the skull. Sorry, this was a different piece that I found. But here are little bits of fossils. So basically you're looking for something that just looks a little different. And so you have to have a very keen eye for it in order to see what looks different. And obviously a perfect situation is in situ, but this just happened to be a fossil find. And I thought it was a really cool demonstration of seeing how you have to spot things. You have to have a keen eye. So then we get to, this is the drama part, because once you found it, now you've got to figure out what it is. Okay, then just an example of an in situ fossil. So this is a specimen that I found in situ. No, not I didn't find it, sorry. One of my colleagues found it. I know it's really difficult to see, but this is actually a skull. And here's the spine. And this had paraloid poured onto it. So I think I mentioned it earlier. So this is like a resin that you pour on top that makes the fossils a little bit more stable because they've been exposed to the elements. So obviously we want to make them as stable as we can so that we can take them out the ground, transport them back to the museum, and then have them prepared. So yeah, this was actually ended up being a beautiful specimen. Here's another example of in situ. So here you can see there's just some bones inside the rock. And again, it's when you first start, it's really tricky to spot fossils because it all looks the same. I found, just to give you an idea of all the bits and pieces. And yeah, again, a surface find. This is fossilous white section over here. And this was a skull that I found, also different. So you're just looking for any kind of exposed bone and it looks a little bit different to your surroundings. And just showing you the process of how we actually extract these kind of specimens when they're in situ. So what we have to do is we have to clear around the fossil, and we have to make sure that there's no loose pieces. So we'll pour some paraloid on and let it dry just to let it stabilize a little bit. And then we end up cutting the rock underneath, and then you end up covering it in plaster like this. You let that dry, and then you flip it over like this. So like I said, this is a skull, and here I've very nicely with, I think, probably in paint, I outlined the skull, just so that you can see, because obviously not everybody will be familiar with these kinds of animals. So this is an orbit. this is a nasal cavity, this is a tusk over here, and then they had these beautiful squamosals at the back that flared back. So once you've flipped it over like this, then you plaster it on top as well, and then you wait for it all to dry and you transport it back to the museum. So the museum, this is what the lab looks like at the museum. And you all work with microscopes, and you have scribes and everything. I'll show you all those bits and pieces in a minute as well, but basically this is what the lab looks like there. And once everything gets transported back, it gets put on the table, and then you get designated fossils that you will then prepare, because as you can see in the previous slide, you can't see exactly the fossil surface and things like that, so it's still covered in rock and everything. So in order for these things to be studied and researched, we have to take that rock off of the fossil. All right, so this is my little setup that I personally have, and I have a microscope, and you use sandbags to prop up the specimens, and obviously it makes a massive mess as well, because you basically, you end up with little bits of rock everywhere and dust. And these are the scribes that I mentioned that we also use to do it. And I've also included a nice selfie just to demonstrate the kind of equipment that we have to use. It is very, very, very dusty. You have to wear a mask. And so when COVID came along, I was kind of used to it, because I was like, oh, well, I do this every day anyway, so I get to wear a mask all the time, just because of all the dust that comes through. And this was a really big specimen that I worked on, or actually still currently working on for Wits. And using the microscope, then you need to wear some eye protection. When you are using the microscope, it's not necessary to use eye protection, just because your eyes are so close to the microscope anyway, so they are protected somewhat from flying pieces of rock, basically. Okay, and this is my shelf that I have, which is full of specimens, and as you can see, it's kind of dusty, and my little mascot on top there that somebody actually made for me, my little velociraptor mascot. So yeah, so a collection of fossils, and as you can see, they just look like rocks. So once they're prepared, then they don't look like that anymore. Okay, then the next part is the suspense of this whole thing. So here is a short, well, it's quite a long video, but I'll only play a little bit of it, of my preparing. I'm just trying to get my mouse to show. Let's stop the pointer. Okay, so here's a little video. I hope I've muted it. If it does come across as noisy on your side, I do apologize. So just be warned that you might have to turn your sound on very quickly. But just to show you what exactly it is, because well, yeah, like I said, I use something called an air scribe. It's just pressurized air that comes through like a pen. And this is what it ends up looking like. So let's see. You can see there's rock, there's the fossil, and there's some rock on top. And all I'm doing is I'm very, very, very carefully taking the microscope. The camera is just angled from the side. So yeah, let's just watch that for a little bit. And because of the way things are fossilized, there is actually usually a layer of separation between the fossil and the rock, because they're slightly different minerals. So yeah, sometimes you do get quite tricky specimens where there isn't melting the rock away and it nicely separates from the fossil. So that just takes that layer off. And can I skip a bit on this fossil? No, just pause. All right, so that's just a little demonstration, and that's the suspense part of this whole thing, is that it's so suspenseful. Like, all I want to do is get these things uncovered just so that I can see what's inside. So yes, so sorry, I think I forgot to mention that this is my part. This is my job, where I do fossil preparation. So I prepare the fossils for research, just so you can see all the intricate details of everything. I am specifically a microfossil preparator, which means that I work on some really, really, really small specimens. So I like to work on very small things that have intricate details, and yeah, but obviously you get bigger pieces and smaller pieces. And so this is what I do. This is my job. Okay. This is something I have also prepared before. It is a Confidon, I think it's called, which is a bit of a funny name, but just to demonstrate that this was in pieces and I ended up gluing it partially together. There are some pieces missing, you can see, but it ended up being such a beautiful specimen. So I have to get in between all these bones and pieces and things so that whoever's researching it can see it nice and clearly, and they can see exactly all the different aspects and the different features of these specimens. Another thing to note, which I thought was really cool and might just be a bit rudimentary in my mind, but these fossils are 250 million years old, and yet the skeleton structure is the same as what mammals have now. And I think that's very cool. Like, there's still spine, there's still ribs, there's still a single bone in your humerus, there's a double bone further down, and the same on your legs. So, and they have the exact same skeletal structure, which I think is very cool. Okay, then another example of things that I've worked on before. This is a Youngina nest. They look like little lizards. Yeah, so this just looks like, this has been prepared at this point. It looks like a huge hot mess, I know. There's a lot of little bones and pieces and that kind of thing. But what it ended up being was a lot of little lizard type creatures that were all accumulated together. So just an idea of, this is the piece, and this is a zoomed in version. Let me get my pointer out again. Here we go. So this zoomed in version over here, where you can see all these little teeth, is actually this little teeth, hopefully. Maybe you can't see on small screens, but yeah, just to give you an idea of, this is what micro preparation is, where you are preparing something so teeny tiny. Oh, there I ended up, I actually forgot that I put a little circle there. So that is demonstrating what you can see through the microscope. And just to give you an idea of scale, I know it covers most of the picture, but that is 1 millimeter. So those teeth are so teeny tiny. This is another different skull in that same specimen that I also managed to find little teeth, and this one actually has a scale bar in it, yay. So yeah, this teeth was so teeny tiny. Let's get the pointer away again. Sorry, a little bit glitchy, there we go. All right, then this is just also to show you, these are, each red circle demonstrates an individual. There were about 25 to 30 individuals in the end on this piece, but this is probably one of my most memorable pieces that I've prepared so far. So yeah, this was loads of fun, and trying to find all the little skulls and also preparing those teeny tiny teeth. So another example of things that I've worked on is something like this. This is a shark tooth. And skulls as well. Like I said, my favorite is preparing teeth, just because they're so beautifully preserved most of the time. And I don't know, there's just something so satisfying about finding all these little teeth. Here is another demonstration of things that I've prepared with a scale bar. So this is what we call mechanical preparation. You do get other sorts of preparation, but we focus on the mechanical preparation here. Now, a lot of people would wonder, but why can't we do CT scanning instead of effectively destroying the specimens? Because this is, destructive kind of, well, it's destructive if you are putting your scribe through the specimen, which don't do that, because these things have survived for 250 million years under the ground. And now we come along and we make a hole in the fossil. That's not a good situation to be in. So, but it's still a form of destructive research. So there are ways that we could CT scan. The problem with CT scanning is that it doesn't always show us what we need to see. And also with CT scanning, you are restricted to the size of your specimen as well. So here is a demonstration of the CT scanning, and let's just play that quickly. This is a CT scan of a specimen, and you can see, can't quite see much just yet, can't see much. I'll just let it play for a second. It's a little bit glitchy because the zoom is taking up a lot of memory on my computer. There you can kind of see something came through on the side. You can kind of see the white spots. And then obviously this bottom right is just a picture, actual picture of what the piece looked like. And there's some more pieces. And so we can see things are showing through, but it isn't always as useful as we hope it to be. I'm just letting it play. And sometimes properties of the fossil and rock are so super similar that it's really difficult to see the difference between the two as well when you do a CT scan. So I just want to let this finish playing. So you could see some white pieces kind of appeared, but as a whole, it was really difficult to see. I know obviously with CT scans, you can take out pictures and things. So what they ended up doing is they had this specimen scanned, and then they decided that they were actually going to get it prepared anyway. So I ended up preparing this beautiful specimen. It's a Brumestiga, and I really love doing the specimen. So the difference between what you just saw of a CT scan is versus mechanical preparation, this is what we could get out of it by doing it mechanically. So you could see it's a beautiful piece. And altogether, it looked like this picture on the left. It was a full species head to tail, and then another species next to it. So the piece that was scanned, sorry, getting that laser pointer again, is this piece here that I'm demonstrating on the right hand side, which ended up being this piece over here. So it definitely was better to have the whole fossil mechanically prepared as opposed to using just CT scanning. And again, beautiful teeth, my favorite. So that is what I do, and you can see all the little indentations on the skull as well. So we have to be very, very, very careful when we are preparing these things. So then we get to the love part of the story. So at Wits, there is this beautiful, beautiful specimen that was found. And so this is a demonstration of a good scan. This was done at the European synchrotron, and it's a Brumastega and a Thrinaxodon. And these two specimens were just called up together. There's a research paper on it. I think you could just Google it if you wanted to. But just to show that these two species ended up curling up together. The one was injured, if I remember correctly. I didn't prepare the specimen. This specimen was, I think, only scanned. I don't think it was prepared. But yeah, just to show that these species of, these animals are so super interesting because these are two different species that are cuddled up together in a little burrow. So just some more demonstrations of things that I have personally done. So here is another, I think this was also a Gomphidon, if I'm not mistaken, on the left. And so I have to make sure that I can even get in between teeny tiny teeth, which is the picture on the right. Here's another picture of species that I have prepared. And again, it's all about getting in between those teeth, getting all the matrix, the rock off, so that those who want to research it can see these species really nicely. So this one on the left was also really fun to do. You can see how, you can also see wear and tear on the teeth as well, which is one of the reasons why I love teeth so much as well. There's so much information you can get from teeth. And here is a beautiful set of digits on the picture on the right that I also prepared. Now, I know I've been talking a lot about paleontology and fossils. This kind of work does obviously apply to archaeological aspects as well. And I have done an archaeological specimen before as well. It was a skull. And this is part of the drama of the story. So the drama here is that on the left, while I was working on this, I unfortunately can't show you the whole picture, but just this portion of it, I knocked off the tooth by mistake. Now, if you are good at what you do, then you are able to repair these things without any visible damage that you can see. So what I had to do was I had to actually fix this thing. And on the right is a picture of the repaired tooth. This piece was so fragile and brittle, and it was really difficult. And it's not fossilized, so it's not a fossil. It was just covered in really hard sand that it like kind of calcified a little bit. So that's how, by me using my methods of my mechanical preparation, that just helped so much. Otherwise, sitting with a little needle or a pick, it would have taken forever. So this is actually a job that I did it ended up being for the Max Planck Institute, but it was something that was found here. So archaeologically, it does apply. My dream would be to work on some hominids that are found in Sterkfontein and the cradle, because I think that would be so amazing. And especially with the micro preparation that I do, I think it would be very cool to work on those sort of species so that they could get some great detail out of it with no damage. So the drama part is if you knock something off, and trying to fix it is quite intense, but you have to make sure that you're good at what you're doing. Okay, so this pretty much brings me to the end of the PowerPoint presentation. I do want to show you some things on my phone just to show you what I've got here in my little lab and show you what my lab looks like as well. But thank you for listening to me rabbit on about Karoo paleontology. And just obviously have to include a selfie that I took that was in Graaff-Reinet at the Graaff-Reinet Museum. There's some really interesting museums out there that incorporate paleontology from South Africa. So I think that's very cool. Okay, so what I'm going to do is I'm just going to share, so you can see there's another participant. Actually, I don't know if you can see if there's another participant, but I'm going to share my screen so that you can see. Let's stop the share here. And let's do this. Okay, so just wanted to show you some of the things that I'm currently working on. Let's turn on the light up here. So this is a little Emidops that we think it might be. And so this thing was covered in plaster. Actually, it's better if I turn the light off, you can see it a lot better. So this was covered in plaster. I had to take the plaster jacket off very carefully and I had to prepare it. Unfortunately, this little guy was not preserved too well, but there you can see there was that foramen that I spoke about earlier, the pineal. And there's the little skull on the side. You can see the orbit. Sorry, I only have two hands. So I'm holding one with my phone and I'm holding the bottle with the other hand. But there you can see an orbit and you can kind of see the face a bit. And there was this beautiful, beautiful hand on the side, which came out really nicely. So overall, a really, really good specimen. I also mentioned that I do work for Wits as well. So this is a piece that one of the dinosaurs that I actually did from Wits. And this is a hand. And so here are the digits. There's one there, there's one there, there's one there, and there's another one here. Unfortunately, we don't have the whole thing, which was real pity, but yeah, this was also a stunning piece to prepare. Like once I got to, there's so much rock to take off, oh my goodness. So once I got to the fossil part, it was so nice. This is the scribes that I spoke about earlier. So what happens is you end up, okay, we're switching hands, hold on one sec. You end up holding it like a pen, and you have to obviously have pressurized air. So I have a compressor that sits in my garage, and the pressurized air comes through here, and you end up being able to take the rock off and using it like a pen. You also get the microscribe. This is one that I actually make myself, which is cool because it's not available in this country. So we do make and sell these, and so you can obviously see it's a hang of a lot thinner, and this enables me to work on the small micro fossils, and it enables me to work on the teeth, and it just, yeah, it's got focus there. So it's got a much, much thinner stylus and a much sharper point. Okay, then I just wanted to show you my shelf of goodies. So, I'm sorry, all the little flumbles sitting there. But yeah, so I have a full shelf of specimens. I've got some beautiful fossils here. They are pitch black. I know you probably can't see it so well through the camera, but they are from Zimbabwe, so these are really nice. This is another piece from Wits that I prepared. This is a birch grove horse, and there's a piece of a rib over there. And yeah, and then there is the other side. Oh, and then obviously here is my microscope setup that I sit and work on all my fossils with. There's some sandbags and my mask. It's a little bit dirty, so it needs replacing. And here's some more fossils. I know they're on the floor, but I have taken them out so we can see them. So this is also from Wits. These are some nice dinosaurs that I'm busy working on. And here is another piece as well, sorry, sunshine, that I'm working on. And if I remember correctly, this is a scapular. I think this is a scapular. And so yeah, so I have to remove all of this rock. It's quite a bit to remove. This is mass matrix removal that I will use with the bigger scribe and take all of this off over here. So yeah, so sorry, I know my picture on the recording is showing me walking away, but this was the only way I could think of sharing these beautiful fossils that I have with me now. Okay, so those are all my fossils, and that is my office. And you can see what I work on. And yeah, so my hope is one day that I can get a bit more involved in the archaeological side of things, and it's just, I guess, all about getting your name out there, because I do independent contracts. So a lot of the work I do is for Iziko, and like I mentioned, I do work for Wits as well, and that's also through the Oxford University. There's a joint project there, and so yeah, so I get to do lots of cool things and just make a mess, because as an archaeologist we really love the dirt, so may as well just make my job about digging in dirt and making everything really dirty. Okay, so thank you for listening, and if you would like any more explanation or any more detail about anything, please let me know. And then just a disclaimer, I'm not a paleontologist. So if there are any bits and pieces that I have not explained properly, it's just because I am not a qualified paleontologist, and getting used to all the names of all these different fossils has taken quite some time. So yeah, it's super interesting, and yeah, it's very cool that South Africa has such a massive deposit of fossils, and there's some really, really interesting research going on out there in the paleontological world. And so, yeah, so I hope you enjoyed my talk, and any questions are welcome.